Good morning, students. Today we are going to see the respiratory system, the physiology of respiration. So physiology of respiration includes the major three topics. One is the mechanics of respiration, how the respiration is taking place, and how the respiration is regulated, that is regulation of respiration. And third, very important, is the transport of the gases. So what is the ultimate aim of respiration? It has to oxygenate the body tissues. For oxygenation of body tissues, we should know about the transport of oxygen and transport of carbon dioxide, which is an excretory product from the cell, and it has to be exhaled. So under these three major topics, we will see the respiratory system and some unusual environments where the respiration gets altered. That also we will see. One in the high altitude. Other one is in the deep sea. So these things we will see in respiratory physiology. So in mechanics of respiration, first we have to see the structures involved for the mechanics of respiration, that is muscles of respiration. So under the muscles of respiration, we have muscles for inspiration and muscles for expiration. So the major muscles for inspiration are the diaphragm and the external intercostal muscle, and the major muscles for inspiration are the abdominal muscles and internal intercostal muscles. Usually inspiration is an active process where work is done, whereas expiration is a passive process. And the muscles of expiration are active during forced expiration. So where we require the muscles of expiration during forced expiration. And we have accessory muscles for inspiration. So inspiration is an active process, and the major muscles are diaphragm and external intercostal muscles. And we have some accessory muscles like ala and sa, sternocleidomastoid and scalene anterior. So these are the muscles for respiration. So the muscles for inspiration, I told you diaphragm. So diaphragm will always move up and down. Okay, so it's a dome-shaped muscle, and when the muscle contracts, the dome shape gets flattened, and it will increase the vertical diameter of the thoracic cage. If this is the thoracic cage, the vertical diameter will be increased. Here the vertical diameter is like this, and here the vertical diameter will be increased. So increase in the vertical diameter. And the other muscle is the external intercostal muscle, and this external intercostal muscle produces two types of movement. One is pump handle movement. So like the handle which moves up the sternum. Okay, so we have the ribs and we have the sternum. So the sternum will move up. This is due to the contraction of the external intercostal muscles. And we have the bucket handle movement. So bucket handle, you can see the bucket handle will be like this, and if this is taken as a rib, the rib is moving in this direction, both sides. So this is the movement of the ribs on both the sides. So this is called bucket handle movement, and this one is the pump handle movement. So the pump handle movement will increases the anteroposterior diameter. It increases the anteroposterior diameter, and this bucket handle movement will increase the transverse diameter. So two diameters are increased. One is the anteroposterior diameter. So this will increase the AP diameter, and this will increase the transverse diameter. So AP diameter, transverse diameter increased by the contraction of external intercostal muscle, and the vertical diameter is increased by the diaphragm. So diaphragm is supplied by phrenic nerve, which is arising from the phrenic motor neurons present in C3 and C4 cervical segment of spinal cord, whereas the external intercostal muscle is supplied by phrenic motor neurons, supplied by the thoracic motor neurons present in the thoracic segments of the spinal cord. And before going for the respiration, how it is taking place, what are the events, we should know there are three pressures in the lung. One is this is the lung, and it is surrounded by the pleura. So one is the pressure between the two pleura. This is the parietal pleura, this is the visceral pleura. Between the two pleura, you have a pressure which is called the intrapleural pressure, which is always negative. This negative is because of the vacuum space between the two pleura, and also because of the lymphatic pump. For these two reasons, it is negative. So the normal value is -2 to -6 mm of mercury. Then we have the pressure in the lung. So that is, we call it as intrapulmonary pressure, or intraalveolar pressure. So this is intrapulmonary or intraalveolar pressure, pressure inside the lung, and this is intrapleural pressure. And the difference between the intrapleural pressure and the intrapulmonary pressure is taken as transpulmonary pressure. So what is transpulmonary pressure? So transpulmonary pressure is a very important pressure, which is derived by the difference between the intrapulmonary pressure and intrapleural pressure. If the intrapleural pressure is becoming more and more negative, it automatically increases the transpulmonary pressure. Say at the beginning of inspiration, the intrapulmonary pressure is 0 mm, that is, it is equal to the atmospheric pressure of 760 mm of mercury, and this intrapleural pressure is -2 mm of mercury. -2 compared to the atmospheric, that is 758 mm of mercury. Now, if the intrapleural pressure is increasing during the inspiration, if this is increasing during the inspiration, say if the value is becoming more negative, say it is going to -6 mm of mercury, and if the intrapulmonary pressure is going for -1, so -1 plus 6, it will be coming around plus 5 mm of mercury, which is the increase in the transpulmonary pressure. So initially, at the beginning of inspiration, the intrapulmonary pressure is 0 minus the intrapulmonary pressure is minus 2. It is plus 2 mm of mercury. So mathematically, so it is increased. That is the value of the transpulmonary pressure during the inspiration. So with this introduction, we go for the events during the respiration. So the neural signals from the brain, reaches the muscles of respiration through the motor neurons, and the muscles of respiration are activated and causes chest wall expansion. This is the first event. Okay, so due to the chest wall expansion, the intrapleural pressure becomes more negative. So normally it is negative, it becomes more negative, and now this causes the increase in the transpulmonary pressure. As I already told you, increase in transpulmonary pressure. So increase in transpulmonary pressure causes expansion of the lungs. So this transpulmonary pressure is a measure of the elasticity or measure of the expansibility of the lungs. So expansion of the lungs takes place, and this expansion causes fall in the intrapulmonary pressure or intrapleural pressure. So now the intrapulmonary pressure is falling, and this causes a negative pressure inside the lung, and the air is pulled into the lung. So air is pulled into the lung. So once the air is pulled into the lung, and this causes the alveoli to inflate. So what is the ultimate target? The alveoli is inflated. Alveoli inflation. So these are the sequence of events: neural signals, muscles of respiration, chest wall expansion, intrapleural pressure become more negative, increase in transpulmonary pressure, expansion of the lungs, fall in the intrapulmonary pressure, and air is pulled from the atmosphere into the lungs because of the negative pressure inside the lung, and the alveoli inflation takes place, and this causes the air oxygen to enter the alveoli. So now we continue. So once the alveoli oxygen is increasing, and once the PO2 of the alveoli oxygen is increased, and there is a diffusion of the gas into the pulmonary capillary. So this is the pulmonary capillary. So this process is called diffusion, and this diffusion is directly proportional to the pressure difference of the oxygen between the alveoli and the capillary. So P1 minus P2 into the area of the alveolar capillary membrane, and inversely proportional to the T is the thickness of the alveolar capillary membrane. If the alveolar capillary membrane is very thick, the diffusion will be reduced. So it is inversely proportional. It is directly proportional to the pressure difference and area of the membrane. So now this diffusion proportionality symbol is removed by using a constant D. So this D stands for the solubility of the gas. So more the gas is soluble, more will be the diffusion capacity. This formula is called as Fick's law of diffusion. Now the oxygen which is entered into the blood, oxygen combines with hemoglobin. So in the hemoglobin, oxygen combines with the heme portion. So heme portion has 4 tetrapyrrole ring. Okay, so each hemoglobin molecule is having 4 tetrapyrrole ring, and it is having 4 ferrous ion atoms. So one molecule of oxygen, hemoglobin will combine with 4 molecules of oxygen. So when one hemoglobin combines with oxygen, we call it as HbO2. So attachment of one oxygen causes relaxation of hemoglobin. So hemoglobin now combines with another molecule of oxygen and forms HbO4. Now this HbO4 combines with another molecule of oxygen and forms HbO6. Now this HbO6 combines with another molecule of oxygen and forms HbO8. So this is the final. So 4 molecules of oxygen is combined with the hemoglobin, and this is called the oxygenated hemoglobin. So the affinity between the oxygen and the hemoglobin is given by a curve called ODC, oxygen dissociation curve. So here we are plotting the partial pressure of oxygen in the x-axis in millimeter of mercury, and this is percentage saturation of hemoglobin with oxygen. So normally at a partial pressure of 26 millimeter of mercury, the hemoglobin saturation is 50%. And if you plot the values between the partial pressure and the saturation of hemoglobin, so you will get a sigmoid shaped curve. Say at 100 millimeter of mercury, the saturation of the hemoglobin is 97%. So 97% of hemoglobin is saturated at 100 millimeter of mercury of partial pressure of oxygen. And if the curve shift, here we have to talk about the shift of the curve. So if the curve is say shifting to the right side, what happens at 26 millimeter of mercury, the percentage saturation is around only, it is less than 50, it is around 10%. That means the affinity between the oxygen and hemoglobin is less when the curve is shifting to the right side. So this is right shift. When the curve is shifting to the left side, what happens at 26 millimeter of mercury, see the saturation is around 97%. That is, oxygen hemoglobin affinity is very strong. So which will be more favorable. When the oxygen affinity is very less, the unloading of oxygen will be good. So shift to the right will cause more unloading of oxygen. Whereas when the curve shifts to the left side, the affinity between oxygen and hemoglobin is very high, so the unloading will be poor. So the delivery of oxygen will be poor. So which shift is favorable to us? It is the shift of the curve to the right side. So what are the reasons that the curve shifts to the right side? So one is increased partial pressure of carbon dioxide, or decreased pH, and exercise, during exercise, and during increase in the during exercise, during increase in the partial pressure of carbon dioxide, decreased pH, and increased 2,3-diphosphoglycerate. It is a glycolytic intermediate, and this, when it is increased, the affinity between oxygen and hemoglobin will be less, and during high altitudes, So in all these conditions, we have an increase in the temperature, where the curve shifts to the right side. So left shift is opposite, where the partial pressure of carbon dioxide is low or increase in the pH, and so these are the fall in the 2,3-diphosphoglycerate. So the curve will shift to the left side. So left shift is increase in the pH or decrease in the partial pressure of carbon dioxide, and fall in the 2,3-diphosphoglycerate. So the shift to the right and left is very important to be noted. Now the transport of carbon dioxide. Carbon dioxide, the major form of transport is bicarbonate. So it is taking place at two levels. So the tissues, from the tissues, the carbon dioxide has to be transported to the lungs, and it has to be exhaled. So this is the thing to get happened. Now what happens in the tissue? The carbon dioxide which is released in the tissue enters the RBC. Okay, so inside RBC, what happens? The carbon dioxide combines with water, the usual reaction where it forms carbonic acid, which dissociates into H plus and bicarbonate. And this H plus combines with the hemoglobin in the RBC, and this bicarbonate comes out of the RBC. Say this is the RBC, the bicarbonate comes out of the RBC for exchange and another anion, chloride anion. So this is done by an exchanger which is called bicarbonate chloride exchanger, bicarbonate chloride anion exchanger. And this protein is called band 3, by a protein called band 3 protein. Now this chloride shift maintains the osmolality of the RBC, and because the chloride is osmotically active, it will imbibe water also and make the RBC little bigger in the tissue, that is in the venous blood. So in the tissue you have the venous blood. So in the venous blood, the RBC size will be little bigger, and this is the reason why the hematocrit, or the packed cell volume of the RBC is higher in venous blood compared to the arterial blood. And in the venous blood RBC, the chloride content will be higher, and this will make the size of the RBC to get bigger, and it causes increase in the packed cell volume, or the hematocrit of the venous blood. Now the bicarbonate comes out in the blood. When it reaches the lungs, in the lungs what happens? The reverse of the chloride shift taking place. So in the lungs, if this is the RBC, what happens? The bicarbonate which is in the blood enters the RBC, and chloride leaves the RBC. Okay, so this is taking place, the reversal of chloride shift taking place in the lungs, in the pulmonary capillaries. And here what happens? The oxygen from the lungs enters the RBC and combines with the hemoglobin. So the H plus which is present with the hemoglobin, it gets dissociated, and it combines with the bicarbonate which is entered and forms H2CO3, which dissociates into CO2 and H2O, and CO2 is exhaled. So loading of oxygen causes unloading of carbon dioxide, and this effect is called Haldane effect. So next we will see the regulation of respiration, how the respiration is regulated. So regulation is done by, first by the centers, it is called the neural control or neural regulation. Second is the chemical influence. The chemicals will give the input, the level of the chemicals. What are the chemicals? CO2, O2, and H plus ions, and some non-chemical influence. Now under neural, you should know about the centers of respiration. So in the pons we have two centers. The first center is called the pneumotaxic center. The second center is called the acoustic center, both sides. And in the medulla we have two group of neurons. DRG, it's called DRG. That is the dorsal respiratory group neuron, which controls the inspiration, which initiate the inspiratory signal or inspiration. It creates the inspiratory ramp signal. And another center is the VRG, ventral respiratory group neuron. It is concerned with expiration. It is normally inactive. During forced expiration it will be active. And these two neurons project into the pacemaker neurons. So these pacemaker neurons will send the signals to the spinal cord and to the muscles. So especially to the phrenic motor neurons and to the thoracic motor neurons. The phrenic motor neurons will supply the diaphragm, and the thoracic motor neurons, it supplies the intercostal muscles. So these are the centers. This centers in the pons, pneumotaxic center and acoustic center. And in the middle we have DRG and VRG, dorsal respiratory group neuron and ventral respiratory group neuron. And we have the pacemaker neurons. Okay, these pacemaker neurons are called as pre-Bolzinger complex. Or pre-Bolzinger neurons. So these are the neurons which generate the rhythm of respiration. So based on input from DRG and VRG. So they are called central pattern generator, CPG. and they are called the pacemaker neurons, and they send the signals to the spinal cord, to the muscles of respiration, diaphragm and the intercostal muscles. So experiment on various levels. When you cut the when you make a cut section above the pons, the respiration, all the centers are intact, so the respiration will be a normal respiration. And when you make a cut between the mid pons, okay, in this section, the pneumotaxic center is a center which usually inhibit the apneustic center. So apneustic center is a center which will always active the DRG, that is, it will activate only the inspiration. So that is practically it is not allowed. So you have the inhibition of the inspiration to be there. So this was done by the pneumotaxic center which inhibit the apneustic center. So when you make a section here, this control is lost. So you will have long inspiration. Okay, inspiration is lengthier. Inspiration is lengthier. And when you make a section here between the pons and the medulla, you make a section here, the respiration will be there, but it is irregular respiration. And when you make a section here, that is below the level of medulla, the respiration is nil, there is no respiration. And here one thing you have to be added, the role of vagus. Okay, vagus is also doing the same action like pneumotaxic center. It will also inhibit the inspiration. Okay, whenever any inflation is taking place, this will activate the vagus nerve, and through the stretch receptors, and the vagus nerve will inhibit the DRG, dorsal respiratory group neuron. Actually, DRG is present in the nucleus tractus solitarius, whereas VRG is present in the nucleus ambiguous. So when you do a mid-pon sectioning where the pneumotaxic control over the apneustic is lost, the inspiration inhibit effect of the inspiration is lost. Then this was done along with vagotomy. When you are doing vagotomy plus this section, what happens? The inspiration already is lengthier, and now the vagotomy, that is vagus nerve, is also cut. So now how the graph will appear here is, like instead of this pattern, the graph will be appearing, prolonged inspiration will be there, and it will maintain for some time. Okay, this is the graph pattern when it is done with vagotomy. Now under chemical regulation, the chemicals will alter the respiration by activating or inhibiting the respiratory centers. So one is the peripheral chemoreceptor, one is done by the peripheral chemoreceptors, where they are located in the aortic body and carotid body. The aortic body is located in the arch of aorta, and the carotid body is located in the carotid sinus, the bifurcation of common carotid artery. So that is the location of the carotid body, and the aortic body is located in the arch of aorta. Now this carotid body contains a cell called glomus cell. So this glomus cell is sensitive to oxygen, that is, when the oxygen level goes down, that is, say in hypoxia, what happens? In hypoxia, the glomus cell, the channel present in the glomus cell, that is oxygen sensitive potassium channel, and this channel is closed, and that causes depolarization or positivity inside the cell, and this causes calcium influx, and this causes release of the neurotransmitter dopamine. Now this release of the dopamine will activate the glossopharyngeal nerve, which is arising from the carotid body, that is the ninth cranial nerve. And this ninth cranial nerve will send signals to the DRG, dorsal respiratory group neuron. So through the ninth nerve, it activates the DRG, nucleus tractus solitarius. Whereas same pattern taking place in aortic body, where the signals are carried by the 10th nerve to the DRG, So the process is the same. So low oxygen level, potassium channel closed, oxygen sensitive potassium channel closed, depolarization, calcium influx, dopamine is released, and the dopamine activates the ninth nerve in case of carotid body, and 10th nerve in case of aortic body, and both these nerves will activate the DRG, and inspiration is activated. So this is the role of the peripheral chemoreceptor. They more respond to the oxygen level in the blood. Coming to the central chemoreceptor, they are located in the near the DRG and VRG. This is the DRG and this is the VRG, and this is VRG, and they are located somewhere near around the DRG and VRG. So this is called the chemoreceptor area, chemosensitive area, or the location of central chemoreceptor. And what is stimulus for central chemoreceptor? It is a level of carbon dioxide. When the carbon dioxide level is increased in the blood, and this carbon dioxide crosses the blood-brain barrier, and once it enters the CSF, in the CSF what happens? The carbon dioxide combines with water and forms carbonic acid, which dissociate into H plus and bicarbonate. Now this H plus is a direct stimulus. For the so this H plus ion, which is a direct stimulus, it activates the central chemoreceptors, and the central chemoreceptors will activate the DRG. So the central chemoreceptors usually respond to the H plus ion in the CSF. So this increase in H plus ion is because of increase in the carbon dioxide in the blood indirectly. So hypercapnia and increase in the H plus ion in the CSF activate the central chemoreceptor. Since it is located very near to the respiratory center, it activates the DRG, dorsal respiratory group neuron, and respiration is activated. Now a note on the lung volumes and capacities. So this was explained by the spirogram. So the spirogram shows the lung volumes and capacities. It is a record of lung volumes and capacities. So what is the first lung volume? It is the tidal volume. Tidal volume. Tidal volume is the amount of air inspired and expired. So we'll start with, for convenience sake, we'll start with 2.2 liter, and it is, the value is 500 ml. So amount of air inspired and expired, inspired and expired, inspired and expired. So the value goes from 2.2 to 2.7 if you add 0.5 liter. So this inspiration and this expiration, either of this is called tidal volume. And then we have the inspiratory reserve volume. So this is the extra volume of air that is inspired after the normal inspiration to the maximum inspiratory level. So extra volume of air that is inspired from the normal inspiration with maximum inspiratory effort. So this value from 2.7, this is 2.7, this takes 2.7, from 2.7 to 6, that is 3.3 liters. This is inspiratory reserve volume. Then we have expiratory reserve volume, that is extra volume of air that is expired to the maximum expiratory level. Say this is the maximum expiratory level. So this extra volume of air that is expired from the normal expiration to the with the maximum expiratory effort. It is around 2.2 to 1.2. This value range is 1.2. So from 2.2 to 1.2, that is 1 liter. And coming to residual volume, that is the volume of air remaining in the lung after maximum expiration. So this is maximum expiration, and this one is the maximum inspiration. So these are the 4 lung volumes. And when you add this tidal volume, inspiratory volume, and expiratory volume, you will get vital capacity. So what is vital capacity? This is the vital capacity. There is volume of air that is expired. The volume of air that is expired after taking maximum inspiration. So this value is called vital capacity. So this is vital capacity. So it is sum of inspiratory reserve volume plus tidal volume plus expiratory reserve volume. It comes around 4.8 liters. Then functional residual capacity, FRC. FRC. What is FRC? It is the sum of expiratory reserve volume plus residual volume. Expiratory reserve volume plus residual volume. ERV plus RV. Or it is the amount of air remaining in the lung after normal expiration. This is the normal expiration. Normal expiration. So that is called FRC. Next, total lung capacity. It is a maximum amount or maximum volume up to which the lung can be expanded, is 6 liters. That is the total lung capacity. It is the sum of vital capacity plus FRC. So vital capacity plus residual volume. Vital capacity plus residual volume. It is total lung capacity. Next one is inspiratory capacity. This is nothing but the tidal volume plus inspiratory volume. It is the maximum amount of air that can be inspired. So this value, from this value to this value, is taken as inspiratory capacity. And this spirogram, this volume is recorded by spirogram, and spirogram cannot measure the residual volume. So the spirometer cannot measure the residual volume. So the total lung capacity cannot be measured by the spirometer. And this record, we call it as spirogram. Coming to the other topics like hypoxia. Hypoxia is deficiency of oxygen at the level of tissues. At the level of tissues, deficiency of oxygen. And this deficiency of oxygen may happen in the atmosphere itself, that is in high altitude. In atmosphere itself, it will be very low. Or when there is any lung disease, when the lung is not properly working. So what happens? The oxygen entry into the blood oxygen level will be reduced. And this type of hypoxia we call it as hypoxic hypoxia. So in hypoxic hypoxia, which is the most common type of hypoxia, the atmospheric oxygen level is low. Atmospheric oxygen level. So atmospheric oxygen level is low, or the lung disease will cause low oxygen entry into the blood. So this is called hypoxic hypoxia, and this can be best treated with oxygen therapy. When you give oxygen, this hypoxia can be corrected. So the deficiency at the level of tissues will not happen. The next hypoxia is anemic hypoxia. Here there is a deficiency of hemoglobin, which is the oxygen carrier. So the blood oxygen is normal now, and the hemoglobin is deficient due to anemia. So you will get hypoxia or oxygen deficiency at the tissue level, or due to some poisoning, especially during the carbon monoxide poisoning. So carbon monoxide is a gas which is having more affinity with hemoglobin. When it combines with the hemoglobin, there is less amount of hemoglobin available to combine with the oxygen. So in anemia and in carbon monoxide poisoning, you will get a hypoxia which is called anemic hypoxia. Here you have to correct the anemia, and oxygen therapy, oxygen at a very high level that can help in the increase the dissolved form of oxygen, and it may have some effect in the correction of the anemic hypoxia. So next is stagnant hypoxia. Here, due to the low blood flow or due to the stagnation of blood flow that happens in circulatory failure or in shock. So due to the poor blood circulation, less oxygen will be available to the tissues. That is called stagnant hypoxia. Happens in the circulatory failure or during the shock. And the next hypoxia is the histotoxic hypoxia, where everything is okay, oxygen is entering into the body, and the hemoglobin is also normal, and the blood flow is also normal, but the tissue is poisoned with some poisons. Say for example cyanide poisoning. So in cyanide poisoning, the tissue will be not in a position to take up the oxygen. So that is called histotoxic hypoxia. Next is a casein disease. Here what happens? So this is a sea level. This is a sea level. When you are sea level. So when you are diver, okay, deep sea diver, when he goes to the depth, what happens here, the atmospheric pressure will be very high. So this may causes collapse of the lung, or it may causes compression over the body and causes restricted lung movement. So the diver is forced to inhale a high pressure gas mixture. So high pressure gas mixture made up of nitrogen and oxygen he has to inhale.